Thanks so much for that lovely introduction, and hello everyone. So we'll start off today's session with a discussion on benefit-risk considerations in drug development. Just make sure. Okay, excellent. There we go. And what I'm going to do is discuss some of the key considerations regarding benefit-risk assessment in regulatory decision making. And we'll really look at the application of these considerations in the form of these case studies. So what I will talk about today are two case studies, which are hypothetical examples, which we looked at and we reviewed in our division. And I'm going to be using these to explain to you how we think about forming these assessments. So before I start, I think one of the things that has to be sort of understood and appreciated is that forming assessments and discussing the benefit and risk of a drug product or a drug development program, this is usually considered a straightforward process. So folks think this is straightforward. You look at the benefit, you look at the risk, and you form an assessment. However, there are times and some situations where this can be complex because it can involve several aspects. So decisions are based not only looking at the benefit, the risk as well, and the disease condition that's being treated by this drug development program. But in addition, it's important to understand that these decisions that are made are not arbitrary. So the agency and the divisions as well as reviewers, we adopt an approach which is somewhat systematic and is really consistent, and that's critical because I think every drug development program is unique. It has unique aspects associated with it, with its drug product, with the population, but the general approach, the overall approach that's taken and adopted has to be something that's structured and systematic, and that's important. These decisions should not be made arbitrarily for individual programs, but should be ones which can be adopted and can actually be used across all development programs. Now, before we go into more detail, one other aspect that should be cleared up a little bit is when we really think about benefit-risk assessment, that terminology, that specific nomenclature is usually reserved for a time when the data or the evidence supporting the benefit of the drug product and the risks associated with the drug product are available. So usually this terminology is used in a situation where we have the data to decide and to make that decision, and that usually occurs at the time of drug approval. So this terminology is usually reserved for a time when we're looking and reviewing data and trying to make a decision about drug approval. However, the same and similar general principles which we use during the drug approval process, the same principles are applicable to decision making throughout the life cycle of a drug product. And this is an important point to take away because we appreciate and understand that as a drug product is being developed from the pre-IND phase through the IND stages, phase 3, approval, post-approval, through the entire life cycle of the product, data will become available. And as more and more data become available regarding that drug product, what we do is we form these, or we look through the data again and again and go through this risk-benefit assessment. And it's not just during the IND phase, but even the post-approval phase. As more data comes in, we again reassess and see what is going on, what's favorable, what's not favorable, and we make these decisions. So again, that's one thing that I wanted to take away is that these types of assessments can take place throughout the life cycle of a drug product. So let's just go into some of the most important and some of the key considerations in this decision making. So as I mentioned earlier, benefit, very straightforward. We're talking about product effectiveness. Now, in the early stages of drug development, you may not have clinical evidence that a product is effective. That's why you're developing this product, right? What you may have, though, is proof of activity. So proof of activity in clinical studies or even non-clinical data that supports the hypothesis that this drug product does or is going to do what you're planning or you're developing it for. So that's in this, from the perspective of this talk, benefit applies to activity, proof of activity, and product effectiveness. Safety is it's a broad term. Obviously includes human safety data. When we talk about safety, we're talking about toxicities that can be seen or that occur in humans in clinical trials or in the post-approval process. But in this context, for this talk by safety, I implied not just safety in humans, but toxicology data. We have to take into consideration other non-clinical data that may be available, or there may be some toxicity concerns which are not specific to the product being developed, but are toxicities which are seen and observed with the class of products that your product belongs to. So class-specific toxicity risk, this is important. And you have to take that into consideration as you're proceeding and thinking through product development, because it's not something that can be ignored. Got to pull that in and think about that. And one of the things, again, important things to take away, which I hope sponsors and folks can appreciate here, is that when there is a toxicity, it's not a showstopper unless you really are in a situation, a tight situation, where there's simply nothing that can be done about this. So again, there are risk minimizing strategies that should be taken in, should be looped in, and you have to think about this. Is this a toxicity that's something that's monitorable? What can be done once this product does get approved to minimize the risk or mitigate the risk of this? So those types of things, you have to start thinking about that. And it could be that the toxicity is something that can be sort of handled or managed, but it could be that it's a showstopper. It could be. So these are the types of things that we wanted to think about. Very important, and you'll see this come up again and again, the disease condition that the drug is intended to treat or prevent. This is key. I mean, and again, it's important for folks to understand that developing a drug product and looking at benefit and risk, it's not in isolation. You really have to take in the setting, the context of the disease, the condition that you're developing this product for, the nature and severity of the condition. And you'll see this again come up. It's an important theme, and take that into consideration as you start thinking about decision making. And tied to the disease condition is the idea of what else is out there. So what else is out there to treat the condition or the indication that you're developing your drug product for? Is this a condition with an unmet medical need? And by unmet medical need, we mean there are simply no available therapies out there. There are no approved products out there. And I give you here an example that our division that we dealt with, where there was simply no approved therapy, for example, Ebola infection. And again, this ties into the Ebola infection example is important. It ties in the previous point that I was bringing up about the nature and severity of the condition. So is this condition that you're developing your drug product for, is it something which is a benign condition with multiple available therapies in the market today, versus a condition which is severe, serious, life-threatening? The Ebola example is one for you, where you know that there's no approved treatment for Ebola infection, and we know from the data that's available coming from Africa, etc., that the mortality rate is high, right? So again, that's a different equation there compared to something which is a benign, non-serious condition. And that's important for you all to take away, because all that information, all these aspects go into the decision-making process. And then at the end of the day, sometimes we have to acknowledge that at the end of the day, you may have a lot of data, you may have less data, but the data is not sufficient. There are still some areas of data gap. There are still some areas of uncertainty, and this becomes very hard. This becomes very challenging, because we all want to be comfortable. We all want all the data to support a decision, right? But at some point, it's not possible to get data, or at some point, it's like we have to sit down and think, like, can we scientifically get some additional data that would help answer the question and reach a decision that we're all comfortable with? And that's an important consideration. Have to sit down. It's sort of incumbent upon us to really sit down and say, okay, what additional information do we need? And sometimes there are situations where you simply don't have data. There is simply no data, and there is still uncertainty about how to proceed, and that's becomes extremely challenging and very, very complex, and weighing that level of uncertainty overall equation. And that's when we really reach out to folks outside, experts in the field. We reach out, we have advisory committee meetings, etc. So again, giving you the whole spectrum of data, types of data, the disease conditions, being pursued, as well as the other therapies available, the unmet medical need, and then finally some level of uncertainty that may still be there. So in addition to these overall general considerations and these general tenets or elements that we look in or we consider when we're making a decision regarding benefit risk assessments, there may be some other considerations which are very unique and very specific to the indication that you're developing your product for. So again, these are just what I've given you so far are just very broad principles and broad considerations, and I hope that y'all can just weave these in and think about this a little bit more as you're developing your product. So I wanted to give you all a little bit more background about the case studies that I'll be discussing, and I want y'all to understand that as Renu introduced, I am a medical officer. I work in the Division of Antiviral Drug Products, which is a division which deals with all antiviral drug applications. So we're looking at drugs, new molecular entities, drug products, as well as biologic products, certain monoclonal antibodies, and those types of products come to our division. We review these, and our division lies within the Office of Antimicrobial Products and the Office of New Drugs at CDER. And as you can see, we review a wide variety of IND applications and NDA, BLA applications. We're looking at a whole lot of infections and infectious disease conditions, not only Ebola, which I mentioned earlier, but certainly a lot of the HIV therapeutics and prevention products come to our division. We have a slew of products for hepatitis B, hepatitis C infection, rabies, and so one of the things is that the case studies that I do present, which are hypothetical examples, some of the very, very detailed and minute issues in those may not be directly applicable to programs which are reviewed by other divisions in CDER, so may not be directly applicable to what you're developing a drug product for. So moving on to the case study, the first case study is one which is, I think we're missing a slide here, so I'm just going to keep going. So the first case study is one which is about an HIV drug development program, and this program was ongoing. The clinical program had been initiated. That's okay, Jeff. I'll just continue. So the first case study, again, very broadly, this is an HIV drug development program which was ongoing, and during phase 2 clinical development, so while phase 2 trials were ongoing, we were notified of a safety concern. And that safety concern was then reviewed by the division, and we looked at all the data that we had, and we made a decision to place the program, the IND, on partial clinical hold. And you've heard these terminologies earlier, so I'm not going to go into details, but partial clinical hold pretty much means that certain individuals can be dosed clinically, but the program has to be sort of truncated, and the development has to be truncated and geared towards the specific population that we're allowing y'all to, the sponsor to dose the clinical product in. So with that, let me just start out with this program. So as you know, for this program, as expected, the prerequisite animal toxicology data had been submitted, and there were no major concerns regarding human safety. The single dose trial, clinical trial, had been initiated, multiple dose trials had been initiated, and again, no major safety concerns. And finally, at this point, the proof of concept data also had been obtained for this drug product. So some proof of clinical activity that this drug product actually does improve the HIV viral load and reduce the viral load in the target population, which is HIV-infected individuals. And so a 6-month trial in the target population was ongoing. This is your phase 2B trial that they were looking at, and we were notified of this case. And I'm going to pause here and let y'all read the details. So as you can see, this is a case of a trial participant, a treatment-naive HIV-infected subject who experiences pretty high increases, actually profoundly high increases in liver enzymes as well as total bilirubin. And in addition to the clinical features and the lab abnormalities, the liver biopsy histopathology is compatible with drug-induced hepatitis. And here you can see very quickly is just the lab parameters, and these are types of information that we actually review. We ask, the sponsor submits this information to us and we look at it, and I don't have a pointer, but I'm gonna ask you to look at time 0, which is when the treatment was started, and soon after treatment was started, you can see the steep increase in the ALT, AST liver enzymes, increases in bilirubin, and then on day 60, when treatment was stopped, subsequently you can see the decline in the same parameters. So the question for y'all today, does this case represent a safety concern for significant hepatotoxicity? Show of hands. Excellent. Yes, this does represent a significant, a serious concern. This is hepatotoxicity. The pathology was compatible with drug-induced hepatitis. And so what are the next steps? What does one do when we see this? In a clinical development program, which is for an HIV indication, important life-threatening condition, and we're seeing this, which is this information, and we are very concerned at this point. So at this point, very concerned. Why are we concerned? Because drug-induced liver injury is really a very serious toxicity. We do not want patients or individuals to have liver toxicity, which ultimately may end up requiring a liver transplant or ultimately maybe fatal hepatotoxicity. And DILI is important. It's one of those things, drug-induced liver injury, which may or may not be dose-related. It may or may not be observed in animals. And it's somewhat unpredictable for different drugs, except for perhaps Tylenol, acetaminophen. And the important thing is this is something that has to be monitored in clinical trials because the incidence can be low. And because of this low incidence, many times DILI is actually diagnosed or identified or identified in the post-approval process after the drug has been approved and is being marketed. That does happen. And so we keep our eye open for a case of DILI. It's very important once we recognize this that we take action. And in this case, the reviewers, the review division, we looked at this one case, and we asked the sponsor for additional cases. So do they have similar cases in their clinical development program to date? Would they have any other cases of abnormalities in liver enzymes, abnormalities in total bilirubin? We wanted to see that information. And the sponsor did respond back to us in a very timely manner because they understood the urgency of the situation, and they sent back to us reports of 10 cases, of which 4 cases were identified and were considered clinically relevant. And we looked through these cases, and we realized that these cases basically were occurring at all doses, and there was no one dose that was kind of associated with toxicity, but it was sort of all over. So it looks like I'm missing another slide here, but I will keep going. So the first case that we saw was a sentinel case, which I described to you. The second case was somewhat confounded because the individual was co-infected with hepatitis B. A third case was compelling because it had de-challenge and re-challenge information. And finally, there was a fourth case, which was also somewhat confounded, but nevertheless, the totality of data really, along with that first sentinel case of biopsy-proven DILI, was very compelling. And I want to take the time here because this slide is actually showing, I'm going to take time here to discuss with you how the review division and how the reviewers approach the situation when they see any toxicity. So not just drug-induced liver injury, not just hepatotoxicity, but for any toxicity, this is our overall approach. We really look at the cases very carefully. We actually go through the narratives of the cases. We want to see the information. We want to understand what was the timing of onset of this toxicity relative to when the drug treatment was started. Was there resolution or diminishing lab values or improvement or de-challenge when the drug was stopped? What are the risk factors? Were there a certain subset of people that this was occurring in because they had a certain risk for development of this toxicity? And were other conditions or other causes, for in this case we're looking at liver injury, were there other causes of liver injury that were nicely evaluated by the investigator, and were they excluded? Did the person have viral hepatitis, alcoholic liver disease, etc.? Because that helps us understand if this was a clean case versus a case which is very much confounded. What were the other drugs the person was taking in this particular case? We were looking for other hepatotoxic drugs that these individuals may be on, that subjects may be on. And then again, very important, what is the track record? Do we know whether drug-induced liver injury in this case is something that is associated with this class of compounds? That is important because it gives us an idea of what to expect, and our threshold for just being more alert, and a threshold for taking into consideration whether this was serious or whether this was something that was just maybe just one odd case, maybe it was really not related to the drug product. And so getting that additional information is very important. And finally, re-challenge data. If there was any subject that was actually re-challenged with a drug product and they again developed the increase in liver enzymes in this case, or the drug toxicity, all that is important, and all that's pulled together and weaved together again to try and understand what the causality is, making a causality assessment. So back to this case study. So at this point, again, I mentioned to you, HIV is it's a serious life-threatening disease condition. At the time when this IND and this case study was this IND was presented to us, or this issue was presented to us, there were very few HIV drugs that were approved and were in the market, and very few classes of drug products had been approved. I think there were 3 drug class products that had been approved, and this was taken into consideration. We looked at all the information in terms of the trials that were ongoing. We looked at the number of subjects that had been dosed. And with HIV, one of the things that you have to understand is there are 2 discrete subpopulations of patients, treatment-naive patients, and then treatment-experienced patients. And treatment-experienced patients, the only thing you have to remember really is that treatment-experienced patients have fewer treatment options left because they've sort of burned through some of the earlier regimens. And so in this population here for this particular product, there had been at least 300 treatment-naive patients or subjects that had been exposed to drug product for a median time of 10 to 12 weeks. And there were fewer treatment-experienced subjects enrolled in a separate trial. And when we looked at the incidence of increases in grade 3 or 4 ALT or AST, as well as the total bilirubin increase, we noticed that these cases were all in the treatment-naive patients, and there were none in the treatment-experienced patients, although we acknowledged there were very limited data really available for the treatment-experienced pool of patients, just 50 subjects treated for about 30 weeks. So we took that into consideration, and this is sort of, at the end of the day, how we processed all the information and how we reached the regulatory decision. And I don't want you to read through everything, nor will I read through the whole slide point by point. The idea here is to emphasize that there are several considerations, starting with the intended indication. As I mentioned earlier, HIV is a serious disease, and at that time, when we were dealing with this case, there were fewer treatment options that were available to the treatment-experienced HIV-infected patient population. And this is primarily because HIV is one of those diseases where the virus can mutate and resistant substitutions develop, and folks need a regimen which involves 2 drugs from one class and another class. So it's a combination regimen. And so there's something very unique about HIV and the need which for this patient population. And so we kept that in mind. We knew that this was a drug that, even though investigational, this does work. There was clinical proof of activity data. There were viral load declines which were observed in patients. And so we knew it worked, but we were very aware of the hepatotoxicity concern, right? At the same time, we took into consideration, kept in mind that this is something that can be monitored. And so we allowed the sponsor to actually proceed with clinical trials in the HIV treatment experienced population, provided that they had extremely stringent and very conservative criteria in terms of monitoring the patients, as well as in terms of analyzing the safety data, and then also reconsenting the individuals for participation. So this was the approach we took. But again, the idea was to carve out a certain population in which dosing was still acceptable in a clinical trial setting, in a very monitored setting, and again to allow drug development, but in a very, very structured fashion, so we could actually look and detect for this toxicity, because it is a concern. So that's where the decision making was sort of employed, taking into consideration everything, and a partial clinical hold decision was taken. Before I move on to case study 2, I'll just stop because I know there were a couple of slides with the first case study that were missing. Does anybody have any questions? Yes, go ahead. We have to repeat that question, yes.
For the patients who were treatment naive, who were on the drug in the study, were they then terminated from the study?
That's right. So the treatment naive studies were on, so the IND was on partial clinical hold. The hold was such that it allowed dosing only in treatment experienced patients. And so that meant that any other population, including treatment naive patients, could not be dosed under the IND. And that was because of the data that supported dosing in treatment experience and only treatment experienced patients. So that's a good point you're asking, because that means no dosing anybody except those folks that the hold allows dosing for, which was treatment experience. So and when you look at the hold letters, and hopefully you will never get a chance to look at hold letters from us, but when you look at them, they're very specific, and they not only include information about who can be dosed and who cannot be dosed, but also about how it is that a sponsor can kind of get out of hold in terms of what additional information would be needed in case they want to pursue the other population, for example. What additional information is needed? And in some cases, like this case, there was really nothing that could be done in terms of gathering or obtaining or sending us additional information to change our decision. I mean, the partial hold for the niche population, that's pretty much all that could be done. And the sponsor did do that. So now just keep in mind that I haven't given you all the details of the case. I just as an example, so over here, so I've kept some of the details away. So maybe I just don't want to share excessive information with you at this point, because again, just want to keep it as an example. So moving on to case study 2. That's okay, Jeff. I know the slides aren't working, but so moving on to case study 2. This one is a similar type of situation, but with a difference. Here, the clinical program was one where clinical dosing was taking place. This was for an indication of herpes, treatment of herpes infection. Clinical dosing was ongoing, and during phase 2, we were notified of an animal toxicology concern. So toxicity in an ongoing animal study, and what did we do? And what was the thinking process behind what we did? So again, a hypothetical scenario, the drug product is being developed for the treatment of genital herpes in immunocompetent individuals. As the majority of us know already, this is a sexually transmitted infection, and genital herpes infections in otherwise healthy individuals, these are cold sores. They're infections which occur, which recur, but they're really not viewed as life-threatening. They're bothersome, they're self-limited, but not a life-threatening condition. However, in some individuals who are immunocompromised, like transplant patients or HIV-infected patients, they can have a much more serious and much more severe disease course with severe ulcerative lesions, etc. So keep that in mind. And then, in general, for herpes, there are several treatments which are approved, and they're reasonably well tolerated. But again, for that group of patients who are immunocompromised, who have severe disease, and who may have resistant herpes simplex virus, there are very few drugs that are approved. In fact, just one drug that's approved, and that one does have significant toxicity associated with it, including a boxed warning. So the drug is being developed, investigational agent being developed for the treatment of genital herpes in immunocompetent adults. It has a novel mechanism of action, and so it exerts antiviral activity at a very different site from the currently approved drugs. So it's somewhat different, unique mechanism of action. And the initial toxicology package for the initial IND, when clinical studies were initiated, was not concerning. The new IND was allowed to proceed. And as I mentioned earlier, it was during phase 2 clinical development that the FDA, the agency, was notified of animal toxicities which had not been identified previously. And the concern here is these were severe drug-related toxicities, which was seen in a chronic toxicology study, a long-term chronic toxicology study, and it resulted in some unscheduled animal sacrifices. So this was concerning to the division. This was concerning for a variety of reasons, but when we get data from toxicology studies, from toxicity studies about information or drug effects in animals, and I know Hanan's gone through this yesterday, Dr. Guntus discussed these yesterday with you, our reviewers really go into details again and look at what was the toxicity in animals, and with that, what is it actually relevant to humans? So what are the target human concentrations? When relative to the concentrations at which the toxicity occurred in animals, and what's the NOEL, what's the safety factor, what are the safety margins? Was it in one species or more than one? So how serious is this toxicity? And our pharmacology, toxicology reviewers discuss this with us, and we look at it. And one of the things, this is important because once you start dosing in humans, you really want to go into clinical trials in humans looking and bearing in mind the toxicology profile. So if there is something of concern, you really want to gear or design your trials so that you can actually detect that toxicity in animals and characterize it in humans. And what we usually do when we have, sometimes we're made aware of toxicology or drug toxicities in animals, and we are very concerned about it. Sometimes what we do is in first-in-human trials, we start slow, and we ask the sponsor to start with dosing a small number of subjects. We say sometimes, dose 1 or 2 subjects, stagger dosing between subjects for a specified interval. So dose a couple of subjects maybe in a week, no more than 2 subjects a week, for example, and sufficient monitoring for these initial dose administrations is something that we like to see, because we want to make sure that the initial subjects were dosed, what happens, and are they being carefully monitored? This is important. Also, the protocol, the first-in-human trial, includes and should include some stringent stopping rules for subjects. So if there is a certain type of toxicity, then the next dose is not given. A certain threshold, like grade 3, grade 4, clearly do not redose that subject. As well as conservative criteria in terms of stopping rules for the entire cohort and trial. So these are some of the things that we think of in the first-in-human trial. Stringent enrollment criteria. And our division, we have several monoclonal antibody products, and for these products, we know with monoclonal antibodies that infusion reactions, for example, may occur because these are usually parenterally administered infused antibodies. And so in these, for these new INDs, we actually tell the sponsor, or we try to make sure that the protocol includes sufficient monitoring for infusion reactions. In some cases, for the first-in-human trial, for the first few subjects, maybe even 24-hour monitoring in a monitored setting, so that we can capture these events and ensure human subject safety. And in some rare instances, when we really are concerned about toxicity, we even ask that the trial have a medical monitor or a data monitoring committee which can oversee safety independently, so that subjects are dosed in a manner that's acceptable and that's really safe for human subjects in a trial. So for this product and for this situation, basically, our decision was to place the IND on full clinical hold. And full clinical hold here means no dosing. You cannot dose any subjects, any human subjects, or any subjects in clinical trials. And why was this? Because we took into consideration not just the toxicity in animals, that animals were dying and were being sacrificed early, but also all the other issues that went with this drug product that was being developed. So it was being developed for a relatively benign indication, genital herpes, which is self-limited. There were alternative therapies available. Although there was some prospect of benefit, the risks clearly outweighed that prospect. And so this IND was placed on full clinical hold. And as I mentioned earlier, in our clinical hold letters, we talk about the population or the hold restrictions, and we also talk about or describe what additional information could be submitted by the sponsor, could be gathered by the sponsor in order to come off hold, and what's the path forward. And in this case, sometimes we advise the sponsor to really just reevaluate their development program and just completely, maybe just think ahead and just revise their development program. And sometimes that's the only feasible approach, where they just change the patient population or they just revise the entire program. And then some of the other approaches. So we discuss all this in our clinical hold letter. And in this case, the program did get revised, and the sponsor did take up on our advice, and they did revise the program so that the target patients who they chose to now develop this drug for were immunocompromised patients with resistant virus. Again, niche population in need for a drug which is perhaps less toxic, and this was an investigational agent with from a new class, so different class with different antiviral mechanism of action. So that was again something that was really going for this drug product, and so that's how they chose to move forward. But again, it was important for them to carve out and carefully design their clinical trial, even moving forward, even in this niche population. We wanted them to monitor very closely, very carefully, and ensure that the doses that they were administering were not the doses which were associated with toxicity at all. So just very, very fine-tuned and very refined approach in terms of what's acceptable and how they can move forward. So it looks like I'm running out of time, and I'm going to go through the rest of the slides very quickly if they appear. So yes, so as I mentioned, they considered, we advised them, and the sponsor considered dosing for the revised population. Yes, so again, they limited their exposure to defined dose and duration, and then there were some other issues that we took into consideration. But again, the recurring theme here is, when there is a concern, gotta be careful, right? Gotta go proceed very, very conservatively, very, very stringent toxicity monitoring, conservative toxicity management plans, conservative stopping criteria, and frequent periodic assessments of safety. I mean, this is what we want to see. This is what you want to see, because we really don't want to expose subjects in clinical trials to unacceptable toxicity. And this was a regulatory decision for the second time around when the sponsor came in, and we allowed them to proceed with developing the drug for the very limited population. And so the full hold was then converted to a partial clinical hold. Partial as in we can only focus on that one niche population. Again, we'll not go through the details of this, but to give you an idea of how comprehensive our overall assessment is, and it takes into consideration everything that's known about, sort of everything that's in the universe in terms of that product, the class of products, the disease, the condition, as well as what other agents are available, and what's the medical need. So to summarize, context matters. Safety, toxicity concerns are not assessed in isolation. Really, you're looking beyond just what data is available for that drug. We're looking above and beyond that. We really are looking at the totality of data. It's the aggregate findings. It's the aggregate assessment that we have. It's not just a disease condition. We take into consideration severity of illness, the patient population, all the available therapies, how well this treatment works, how well does the product work, what is the side effect profile, are there risk minimizing strategies, and sometimes there are considerations even beyond that. Drug interactions. Is this something that can be feasibly, like a drug product like this, once it's marketed, it's approved? Are there important drug interactions, for example, with oral contraceptive pills or some other very important drug product that's very commonly used? Everything, sort of has to gel in and make sense, and so we take that into consideration as well. So again, just to end with that one thought, that benefit risk assessment is something that's done at the approval stage when all the scientific evidence is available to make the decision. But these types of principles and these key considerations are the tenets of, or the key elements of how reviewers, the division, and the agency really think about regulatory decision making. And this happens throughout the product life cycle. It's not just at the approval stage, but it's a continuous process. So with that, I thank you for listening, and I'll go ahead and take any questions you have.
We're going to run questions a little bit differently to keep us on schedule. For those of you online, if you have any questions for Charu, please submit them via email to cedersbia@fda.hhs.gov and mention Charu's name, and we'll make sure that they get routed to her. For those of you in the room, just like yesterday, Charu will be one of the presenters who is going to be meeting with us at the end of the day in those one-on-one small gatherings, just like we had yesterday. So if you have any questions for her, go ahead and jot those down.